seminar 13, Gel electrophoresis. Gel electrophoresis. Gel electrophoresis is a technique that uses gel, a thin lap of gel-like material, as a molecular sieve to separate nucleic acids or proteins on the basis of size of electrical charge. To separate, identify, and purify nucleic acids or PCR products use two methods of gel electrophoresis: agarose gel electrophoresis and polyacrylamide gel electrophoresis. How gel electrophoresis would be used to separate the various DNA molecules in the three different mixtures. A sample of each mixture is placed in well at one and the end of a flat rectangular gel. Agarose gel preparing, running, and staining agarose gels. Usually used 1 to 2% of agarose concentration for gel electrophoresis. Agarose gel treated with ethidium bromide for later visualization of DNA amplicons. Final concentration 0.5 microgram to milliliter. We place the agarose gel into chamber and add enough electrophoresis buffer added to cover the gels to the depth of at least 1 millimeter. Cover the gels at least 1 millimeter. Make sure each well is filled with buffer. Make sure each well is filled with buffer. Then carefully place it by the pipette over a well and gently expel the samples. Carefully place the samples by use pipettes. The sample should sink into the well. Be careful to not puncture the gel with the pipette tip. Then place the cover on the electrophoresis chamber and connecting the electrical leads. Connect the electrical lead to the power supply. Be sure the leads are attached correctly. DNA migrates towards the anode. From cathode to anode migrates the DNA samples. When the power is turned on, bubbles should form on the electrodes in the electrophoresis chamber. A negatively charged electrodes from a power supply is attached near the DNA containing end of the gel, and the positive electrodes is attached near the other end. Because DNA molecules have negative charge owing to their phosphate groups, they are all travel through the gel towards the positive pole. As they move, a thicket of polymer fiber within the gel impedes longer molecules more than it does shorter ones, separating them by length. In this picture, you can see the migration of DNA samples from cathode to anode, DNA migration direction. Wells and the samples wells and samples of DNA migrated to anode with DNA samples with bromophenol blue staining. After the current is applied, make sure the gel is running in the correct direction. Bromophenol blue will run in the same direction as the DNA. We'll add DNA samples and the DNA markers loaded to the wells, into wells. DNA ladder standard loaded to the into wells. Bronphenol blue migrates at approximately the same rates as 3,000 base pairs DNA molecule DNA migration. Bronphenol blue ladder DNA ladder migrated in the same direction. Inclusion of DNA ladders, DNAs of null sizes on the gel makes too easy to determine the sizes of unknown DNA's molecules. DNA ladder standard added to gel, well of gel for determining the sizes of unknown DNAs. Visualization DNA samples. For visualization of DNA samples, we added to agarose ethidium bromide. When we prepare the agarose gel, we add the ethidium bromide. For further later visualization of DNA samples. DNA ladder in the first well, and the next is the samples of DNA, and the DNA ladder and at the end of the gel. Wells and the PCR products, we can see the DNA samples separated by the size and produce the bands of each product, 500 base pairs. The shorter samples of DNA in the bottom of the gel separated. Thus, the gel electrophoresis separates a mixture of linear DNA molecules into bands, each consisting of DNA molecules of the same length with shorter molecules towards the bottom. Agarose gel electrophoresis usually used 1 to 20% of agarose gel for separate DNA molecules. For DNA molecules, separation usually used 1% agarose gel, and for separate of PCR products, used 1.0 and 2.0 gels. Recipe of preparation 2% agarose gel, 2 grams, and added ethidium bromide, and deionized water. For agarose gel electrophoresis, used horizontal gel electrophoresis chamber. And second method, polyacrylamide gel electrophoresis. Polyacrylamide gel electrophoresis consists of 2 gels. Polyacrylamide separating gel at the bottom, and 5 polyacrylamide, 5% polyacrylamide stacking gels. This recipe of preparation of stacking gels and the separating gels. Separating gels consist acrylamide, bisacrylamide, tris, ash chlor, and SDS, deionized water, trimate, ammonium persulfate. Final value 7.78 milliliter. And the stacking volume, stacking gel contains the same chemicals and reagents, and the final volume 2.52 milliliter. In polyacrylamide gel electrophoresis, uses vertical DNA electrophoresis chamber. Control question: gel electrophoresis and the DNA standard. tell about the DNA standard in the way should you prepare for last laboratory lesson these questions, and we can continue our lesson and watch the video about agarose gel electrophoresis. Agarose gel electrophoresis.
This video will take you through the process for loading and running DNA samples on an agarose gel. You will be using a mini subcell to perform agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode of each. This video will take you through the process for loading and running DNA samples on an agarose gel. You will be using a mini subcell to perform agarose gel electrophoresis. The subcell has an electrode wire running across the bottom of each end. This provides for an electric current to pass, which separates the DNA fragments in the samples. First, align the gel so that the wells are closest to the negative or black electrode. DNA is negatively charged and will move from the negative electrode through the gel toward the positive or red electrode. Place the agarose gel into the gel chamber. Add electrophoresis running buffer to the reservoirs at each end of the gel chamber, and keep adding buffer until the wells in the gel are covered by at least 2 millimeters of buffer.
Buffer covers the gel up to 1 millimeter.
Place your samples to be loaded in the correct order according to the lanes they are assigned to be run in. Check your lab protocol for this information. To obtain a DNA sample, slowly depress the plunger on your adjustable micropipette to the first or soft stop. Holding the plunger down, insert the tip of the pipette into the microtube with the DNA sample. Place the tip close to the bottom of the sample, then slowly release the plunger button. When loading the samples, keep the pipette tip perpendicular to the row of wells. This will reduce the risk of accidentally puncturing the wells with the tip. Lower the tip of the pipette until it breaks the surface of the buffer and is located just above or just inside the well. Slowly apply pressure to the plunger button. Observe as the sample fills the well. Make certain you stop at the first stop on the pipette, pause, then slowly remove the pipette while keeping your thumb down on the plunger. Once all the samples have been loaded, avoid any bumping or movement of the gel chamber. This might result in the samples spilling into adjacent wells. Place the lid on the gel chamber with the terminals correctly positioned to the matching electrodes on the gel chamber, black to black and red to red. Connect the electrodes to the power supply, again making certain the electrodes match the terminals on the power supply, red to red and black to black. Switch the power supply on. Then set the correct constant voltage for running the samples. See your lab protocol for this information. If a timer is available on your power supply, set the clock for the proper time for your run. Press the start button to begin the flow of current that will separate the DNA fragments. this point, you should be able to see bubbles coming from the wires at each end of the gel box. Notice that the positive or red electrode has fewer bubbles than the negative or black electrode. In a few minutes, you should see the samples begin to migrate from the wells into the gel. Notice as your DNA samples run, the dye front moves from the negative electrode towards the positive electrode.
Goodbye, students. Our lesson finished.
Goodbye.
Goodbye.